So next we are starting with dynamics of disease transmission. Dynamics of disease transmission means how is the disease being transmitted. So disease transmission refers to mechanism by which the infectious agents is spreading from one host to the another. Host means the person or the human being. Then the transmission begins with the presence of reservoir or the source of infection, means there should be some source of infection which spreads, and then it goes inside the susceptible host. Then this process forms the foundation of chain of transmission, and it has three links. Three links means there should be one reservoir of the disease agent, it should multiply, and it should be in multiple number. Then the mode of transmission, how it is traveling to the host, and the vulnerable host. Vulnerable host means who is receiving the agent, that is the human being here. So understanding all these three links is very important when we are when we have to interrupt the disease, and when we have to break the chain of spreading the disease, right? So first step in the development of communicable diseases, reservoir. So we'll come directly into to the point that what is the reservoir, or what is the resource of source of infection. These are certain definitions. So source of infection can be the person, can be the animal, object, or the substance from which the infectious agent is being passed on to the host. Then the reservoir can also be defined as any person, animal, or arthropod, parasite, plant, soil, or any substance in which the infectious agent was living and multiplying, and it is exposed to the host, and then it reproduces itself, and how is it is transmitting to the host. So it is important to note that the source of infection and the reservoir are always not the same. Like for example, if we talk of hookworm infection, that is the worm infestation in our gut, the reservoir is man, but the source of infection is contaminated soil. But when we talk of tetanus, the reservoir and the source both are same, that is the soil. So here we'll divide the reservoirs into three categories, that can be human, animal, and the non-living things. So human reservoir means the human body, which is the reservoir of the disease agents which is causing the disease to some other human being. The person, the human being is causing disease to the next human being, right? So person may can be served as a case, he can be actively showing the symptom, or he can be the carrier of the disease agent which is silently harboring the disease agent and transmitting the infectious agent without any clinical sign. Means the human being himself is asymptomatic, means usko koi symptom nahi ho raha hai, he doesn't know that he is having the disease, and still he can transmit that infectious agent to the another person, right? So this is very dangerous uh, stage when the person himself is not having any symptom, and he doesn't know that he is carrying the infectious agent within his body and can be infectious to another person. So there can be direct, or there can even be indirect human to human transmission. Means the person one who is asymptomatic, if he is, if the bacterial infection, fungal infection, or any viral infection goes from his used things to another human being, so that is the indirect human to human transmission. So here we is the classification, like what are the cases and what are the carriers. Cases can be the primary case, index case, or the secondary case. We'll study it in detail. uh, the carriers can be incubatory, which are asymptomatic, convalescent, or the healthy, and it also depends on duration of the disease agent, temporary है, chronic है. What are these? uh, how is the disease agent coming into the human reservoir. But then what is the portal of exit? Is it urinary, intestinal, respiratory, or any other source of infection? We'll talk of cases in detail. Cases means the person in the population, or the person who has been identified as having the particular disease or any health disorder or any other condition which is under investigation. In epidemiology, we do the research, right? So when we are researching, we are talking of case, and the case is the person who is having the particular disease, and that disease which is being under investigation by the epidemiologists. So cases can be of means they can be identified using three criteria, like what are the clinical signs and symptoms that are caused to them, yeah, what are the symptoms coming to them, then biochemical markers, that are the lab tests and the lab investigations also, right? So infections within the host may be present in different forms. It can be latent, subclinical, symptomatic, asymptomatic. It can be any, right? So different types of cases based on disease expression. If we talk of this, illness may range from mild, moderate, typical, severe, and fatal, but in epidemiology, mild cases may pose greater public health risk than severe ones. Why? Because severe ones are isolated. They are not means they are not socializing. They are not meeting people. They are not having any direct contact with the other person. But the mild cases are mobile. They are socially active, and they can uh, transmit the disease, right? So whatever we said, yes, severe cases often bedridden, so they are limited, having limited contact, they do not spread the disease. When we talk of subclinical cases means hidden infections are there. Hidden infections means no visible symptoms are present, but the agent is multiplying in the host's body. So, despite the absence of symptoms, he can shed the disease or the disease agent into the environment, and from the environment or from the contact, he can transmit that disease to another person. Means the chain of transmission is there, but uh, the other person is having the symptoms, but he himself is not having the symptom. So that is a subclinical case. So whenever we have a certain communicable disease, we explore his environment also, right? So detection has to be specialized, and we have to see that what is the antibody response, what are the biochemical assays, what is the skin sensitivity, and how can the organism recover. Then is the latent infection. Latent infection means the infectious agents remains dormant within the host, no shedding, but he is having the disease. He is having the infectious agent. It is in dormant state. He is not shedding the agent. He is not having any symptom, and the infection is not transmissible during latency. Latency means the dormant stage of the agent. Whenever it will be active, whenever it will have the active environment, it will start shedding. So this latent infection can become the subclinical, it can become the clinical case anytime. So when we talk of classification, so there can be the primary, index, and secondary cases. Primary case means the first case of the communicable disease in the population, means the first human being who has been detected by the disease. Index case is investigators when notice the first case, that is the true primary, which cannot be the true primary case, or it can be the primary case, right? Secondary cases which who develop the disease after the exposure to the index or the primary cases. So these are certain terms that we use while classifying the cases. Then transmission risk from cases. The transmission risk means the clinical or the subclinical person can be the source of infection, and the infection or the pathogens may exit from the body through stool, vomits, coughing, sneezing, saliva, and any other secretion of the human body. Next, we talk of carriers. Carriers means the infectious agent may not be fully eliminated from the body. Means there are certain limitations where the infected person is harboring the disease agent in the absence of any clinical symptom. Any clinical symptom means he is asymptomatic. No, he is asymptomatic, but he is a potential source of infection. So, what are the components of carrier state? The person is considered a carrier when he met the disease agent in the body, but there are no signs and symptoms of illness. Then the agent is shedding through the discharge, but he is not uh, aware of it, but he is posing the risk to others. So while we classify carriers, we can classify them by type or we can classify them by duration. So if we classify the carriers by type, we can say, so if we are classifying the carriers by type, they can be incubatory carriers, they can be convalescent A carriers, or they can be healthy carriers also. Incubatory carriers are those which are shedding the infectious agent during the incubation period. Means they are not having the symptoms, but still they are having the disease and they can transmit the disease unknowingly, right? They are, then these diseases are common like measles, mumps, polio, pertussis, influenza. Convalescent carriers, they are shedding the disease during their recovery stage. Means they have developed the symptoms, they are having the medicine, but sorry, not but they are having the medicine and they are in the recovery stage. Means they are having the treatment and management of the disease, but the disease has not gone, means the infectious agent has not gone from his body completely. So they are also a threat to close contacts and they require post recovery surveillance also, like what we were doing in COVID. We were having the post test also, that whether the people are COVID positive or negative after their treatment and management. So what next are the healthy carriers? Healthy carriers means subclinical infections are there. They are not showing any symptom, but still they are harboring the disease agent and shedding the infectious agent. So it can be seen in polio, cholera, meningitis, diphtheria, and salmonellosis. So these are certain diseases where we can have the healthy carriers. Healthy we are saying because they are not having any symptom, and carriers are because they can shed the disease agent. Then when we talk of classification by duration, means there are temporary carriers and they can be chronic carriers also. Temporary means they are shedding the agent, disease agent, for a very limited time. They can be incubatory, they can be convalescent, or they can be healthy, but they are shedding the disease for a very limited period of time. Means they had the symptom, they recovered from the disease, and the disease agent is not present in their body completely. But there are chronic carriers also. Chronic carriers means they can excrete the infectious agent over extended period of time. Means particularly very commonly that is the typhoid. Typhoid means you have to take care of your inner self for 15 or till 1 month, and the things like whatever we do, the precautions, we take the precautions, the precautions has to be taken for at least 6 months to 1 year, because typhoid is that disease that which can come or reoccur, and it cannot heal within certain or over a small period of time. So we have to see this for extended period of time, right? Then when we talk of chronic carriers, because they are shedding the infectious agent, so they pose a greater public health risk. They can reintroduce the disease into the disease free areas, and very important is that they should be detected and given the treatment as early as possible, so that they are not posing any risk to the public health or any type of epidemic in their area. Then carriers can also be classified by the portal of exit, means the route through which the agent exits the body, like it can be urinary system, it can be intestinal, respiratory, nasal passages, blood, wounds, skin lesions, anything. So these are the carriers who can be classified under this classification also. Next, we have the animal reservoir. Right now, we talked of human reservoir. Human reservoir means human beings. Now, we are talking of animal reservoirs. Animals and birds can also uh, act as reservoirs or the source of infection similar to human beings. They can be active, they can be asymptomatic, they can have the symptom, they cannot have the symptom, they can transmit pathogen without any visible illness, same as what human being does. So this is known as zoonosis, right? And there are over 100 known zoonotic diseases right now which can spread from animal and birds to human beings. It can be through direct contact, it can be through vectors, it can be through contaminated food, water, or the environment. So for example, influenza, it transmits from the birds or the pigs. Yellow fever, it spreads from mosquitoes. Then rabies from the dog bite. So these are certain zoonotic diseases. Malaria, dengue, we all know that. Then what is the epidemiological relevance for this? So we have to uh, understand that what is the disease ecology, how the disease is being emerged. Population should be early detected, it should be early controlled, and it should not be like taking the like the picture of an epidemic. It should not be an outbreak of epidemic. The things have to be under control. So for this, there are certain public health strategies which are vaccination of animals, it can be vector control also, and there is safe handling of livestock and pets. The third one reservoir that we talked of was non-living things. Means the disease agent can be present on the non-living elements in the environment like soil, water, or any inanimate object also. So commonly we have soil. Soil can retain the pathogens for extended period of time. Means tetanus, anthracis, coccidioidomycosis, mycetoma. These are certain soil-borne infections. Water, there can be cholera. The water, contaminated water sources can be the major contributors of outbreak. Then there can be the fomites. Fomites can be present on the surface, any tool, any clothing, any medical equipment, and particularly this is the this is the primary cause of hospital acquired infections. You can say of when we talk of surgical site infections, when we talk of COTI or MRCA. So this is the primary thing, the fomites or the inanimate objects being contaminated or infected, and then it is used in the hospital and causing the disease. Again, what is the epidemiological importance of it? That they play a role in indirect transmission, and controlling measures can be proper sanitation of all the things, sterilization of equipments, safe disposal of waste, and environmental surveillance. So this was the disease transmission.